Macromolecules, structure and function. This is going to be a little bit more in depth about how life uses these macromolecules. So if you remember, we talked about monomers, we talked about polymers. The mono is one, poly is many, and these are the subunits that make up our macromolecules. So there are different subunits for each of the macromolecules that we're looking at. In this case, we're talking about lipids, proteins, carbohydrates, and nucleic acids. When we put these monomers together, we make polymers, which are many, because it's the same sort of a subunit, the same sort of a monomer that's stringed together one after another, after another, after another. And if you remember, the way that these are put together is through dehydration synthesis. So we're making a chain, a polymer. So in this case, we don't know the specific monomer, but we have maybe sugar is the monomer, like glucose is the monomer, and then glucose, or this could be an amino acid, and this would be amino acid. If we have glucose strung together, which one way you'd be able to recognize that is because there is a carbon, hydrogen, oxygen only in most sugars. That would be your carbohydrate. If it were carbon, hydrogen, nitrogen, oxygen, possibly sulfur, then we have an amino acid that would be in this chain. If you see phosphorus, if you see P's, then we have nucleotides, and those would be a nucleic acid in a chain. And then the lipid structure is a little bit different because they aren't chains like this. It would be the glycerol with 3 subunits connected to it, but it's the same idea. Dehydration synthesis is the process that puts monomers together into a polymer. Just a little review of dehydration synthesis, or the condensation reaction. When water forms, it's formed through condensation. We pull water out of the air, water vapor turns into water. That's called condensation. This is the same process by which all monomers are turned into polymers, by dehydration synthesis. Again, to remember, synthesis is building, putting together. Dehydration is taking water out, and then synthesis is putting together. The process causes us to take a hydrogen from one monomer, and the OH, the oxygen and hydrogen, from another monomer, making water, and then we join those two monomers together into a polymer. Hydrolysis is the opposite of this. This is where we break apart a water molecule. So lysis is breaking. Hydro is water. We're breaking water, and then that water getting broken apart, the exact opposite of dehydration synthesis. One of these hydrogens goes to one of the monomers on a particular side, and one of these OHs goes to the other one. So if you remember our amino acids, the H goes on the amine side where the nitrogen is, and the OH goes on the carboxylic acid side where we have a carbon double bonded to an oxygen. It's kind of the reverse of how it's flipped over from how we usually look at this particular monomer amino acid, but it's the idea that the N would be on this side getting the hydrogen, and then the C double bonded to an oxygen would be on this side where the OH would go, or the HO, depending on how you look at it. Okay, so the things you're going to need to know, definitely for a test, and a test will be coming up within the next few weeks, you need to be able to tell me the 4 different macromolecule types. You need to tell me lipids and being able to identify them. You need to be able to tell me carbohydrates, be able to identify them. You need to be able to tell me proteins, be able to identify them, and nucleic acids, be able to identify them. You need to be able to recognize monomers and polymers based on the elements they're made of. So if I see carbon, hydrogen, oxygen only, that's either a carbohydrate or a lipid. And carbohydrates and lipids have very different structures. If I see carbon, hydrogen, nitrogen, oxygen, and possibly sulfur, that's a protein. And if I see phosphorus, if I see a phosphorus anywhere, that's more than likely a nucleic acid. So I would see nitrogen, carbon, hydrogen, oxygen, and phosphorus in a nucleic acid. Now there are, a little bit later when we talk about cell structure, there are some lipids that have a phosphorus stuck to them. They're called phospholipids. Nothing to worry about now, but we will see those also. But for what you need to know now, phosphorus generally means it's going to be a nucleic acid. We also need to know the function. That's what this presentation is primarily going to look at. What does life use these different polymers for? So let's take a look at lipids first. The structure of a lipid is a fatty acid monomer and a glycerol. Now, if you remember, the glycerol is a carbon, carbon, carbon. 3 carbons that each of these one carbon connects to one of each of 3 fatty acids. So we have the glycerol, and we have 3 fatty acids. That makes a triglyceride. Here we see our glycerol, carbon, carbon, carbon. It's connected to 1 fatty acid, 2 fatty acids, 3 fatty acids. Do not forget that the fatty acid, yes, it does have a polar component, but it also has a very long nonpolar component. Most fatty acids are not very soluble in water. They're mostly hydrophobic. Now this is an example of a phospholipid, which makes it a lot more confusing. But what will tell me it's a lipid in most cases is carbon and hydrogen nonpolar bonds. Most lipids are relatively nonpolar. Now, does this have a little bit of polarity here on this side? Yes, but it gets overwhelmed by these long carbon chains. Now, this is a little bit closer look. So the fatty acid is the monomer. Here we see carbon, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. That's our nice long hydrophobic section, meaning it's water-fearing. It doesn't mix with water because of those non-polar carbon-hydrogen bonds. And then I have a polar end, which does have an OH. This is a polar molecule. Remember, in this part, oxygen has a stronger pull on the electrons, pulls them away from hydrogen. So this part, actually, water would be interested in sticking to, but this overwhelms, in most cases, what water is actually able to do. Which means if you have a water, if you have a fatty acid in water, chances are these hydrophobic sections are going to stick together. You'll have a clump, you'll have a blob of fatty acid. You would not have it mixing very well with water. Now, another little side note here. We have a saturated carboxylic acid. Saturated fatty acid, which means that all of the carbons are single bonded to other carbons and they have hydrogens on each side. This is what we call an unsaturated fatty acid. Notice that double bond. That's because carbon's sharing 1 pair of electrons, 2 pairs of electrons with itself, which means there's less hydrogens attached here, which means that the molecule for a unsaturated fatty acid, an unsaturated fatty acid, is a different shape than a fatty acid. What does this mean for the structure? It means that these are very large molecules. Many large molecules at room temperature tend to be solids. So this molecule will lie nicely along another fatty acid molecule and lie alongside another fatty acid molecule. This is very characteristic of saturated fatty acids, and that means they're solids at room temperature. They also lie very nicely inside the arteries of a person, which is how plaques can form, these clogs, and can form in arteries within the human body. Now, unsaturated fatty acids, because of this kink in the tail, they do not lay together very well. As a result, they don't stick together quite as well. They don't form solids like the unsaturated fatty acids do. These are usually liquids at room temperature. They're also good at clearing out people's arteries because this is nonpolar, this is nonpolar. This will still stick. This will still stick to this unsaturated fatty acid will still stick to a saturated fatty acid and could potentially pull this off of the artery and actually help clear someone's arteries out with these unsaturated fatty acids. This is just another way of looking at one of these fatty acids. Now, this happens to be one where we have two different unsaturated fatty acids attached to a polar component. But in all of these cases, we have a polar region, which is hydrophilic, but it's usually overwhelmed by these long chains of carbons and hydrogens, which are hydrophobic. Now, again, this would be something like vegetable oil. It's a liquid at room temperature. The reason why it's a liquid, because the molecules, thanks to this double bond, just do not stick together very well, whereas these stick together very nicely. So this would be a butter, and this would be a vegetable oil. So liquid at room temperature, solid at room temperature. And that's kind of an explanation of how this affects the behavior of fatty acids. Now, the functions of lipids. We do have inside the cell something called a cell membrane, which is made primarily of those lipids on the inside. This is a phosphorus group. So this is a phospholipid. You did see it back here. Again, how do I know it's a lipid? Long chains of carbons and hydrogens. Lots of nonpolar areas. What happens in a cell membrane is this nonpolar area hides in the inside of the membrane, and these polar regions are on the outside. They make up many of the membranes, not just the cell membrane, but they make up other parts of the other membranes inside a cell also. A membrane is a thin, skin-like structure inside of cells, it's used to contain other parts of the cell inside of them. In larger animals, especially mammals, birds also, it does help keep insulation, keeps the body warm. It's also an energy storage molecule. So if an animal is able to make its own body heat, we're talking about mammals, we're talking about birds, we're talking about certain kinds of sharks and tuna, then the fat can actually help keep their body warm. In all animals, it's used as energy storage as well, although like a turtle may have fat. Actually, there is one kind of warm-blooded turtle, the leatherback sea turtle, but so that one gets insulation from fat also. But most reptiles, most amphibians, most fish don't use the insulation. It has insulation to keep their body warm because they don't make any heat inside their bodies, with a few exceptions. Tunas, great white sharks, and leatherback sea turtles are actually able to keep their bodies warm. And part of the way they do that is by having their fat. And this is something that mammals and birds also do. So we find lipids in high-fat foods. Lipids are primarily fats, cholesterol, things like that are lipids. Now, there are some healthy lipids. There are some unhealthy lipids. Like I said before, the unsaturated fats are usually much better for you than the saturated fats. So the unsaturated, this would be in certain kinds of foods. Avocados have certain kinds of unsaturated fats that are good for you. Olive oil has certain kind of unsaturated fats that are good for you. So we find these lipids in those sorts of foods. Fish contain healthy lipids. Meat, depending on the meat, primarily does not have those very healthy lipids in them. Some oils, like olive oil, very healthy. Some vegetable oils, not so healthy. This is a way of starting to look at what your body is made of and what's important for your body to get. So eggs, they have a lot of cholesterol. Cholesterol is a lipid. Eggs are not healthy in large quantities. Small quantities of eggs, fine, but if you eat like a dozen egg omelet, you're not taking very good care of yourself. Nuts also have some healthy lipids in them. That's another thing that you want to make more of your diet a part of. How do we know something is a lipid? If you put it in a paper bag and the bag becomes kind of transparent, that's a good indication you have lipids in it. Not many other substances would do that. This interaction with the paper bag cellulose, not many other materials, not many other substances would do that. All right, now let's look at proteins. The animation got a little weird, but proteins, as we've said before, are made of amino acids. In living organisms, there are 20 kinds. One of the ways that we can tell that all life is related is because they use the same 20 amino acids, from bacteria to humans to trees to mushrooms. All of them use the same 20 amino acids, and the code for them is the same also, which the code comes from DNA. In an amino acid, what you're looking for, now there's two different ways of representing this. This is a little bit more the way that most people would do it. I personally would pop off this hydrogen and put it back on the oxygen. I can discuss in class, but what happens is there's a hydrogen here. When hydrogens pop off, they pop off fairly well. They pop off of acids. So really what's happening is the hydrogen pops off of here and jumps on top of the nitrogen, because nitrogen is not as electronegative. So if you give oxygen enough of a chance, it will pull its electron. It'll pull the electron that it shares with hydrogen. It'll pull it away from the hydrogen. That hydrogen then ends up popping off and sticking onto the nitrogen. I can probably discuss that a little bit better in class, but either way is fine. What you need to look for in an amino acid is a carbon, a nitrogen on one side, a carbon double bonded to an oxygen on the other side, and then this would be an oxygen, generally with a hydrogen coming off. This nitrogen would generally have two hydrogens coming off, although this is an acceptable representation also of an amino acid. Now, this R group can be a whole bunch of different things. We've started to see this in class. It could just be another H. There's an H up here. There could just be another H down here for glycine, which is the amino acid I said we found on the plumes of ice volcanoes on one of Saturn's moons. There may be other amino acids there. We just didn't have the equipment when we sent that probe to Saturn. We didn't have the equipment to say what other amino acids might be in there. As we'll see in our next video, amino acids are relatively easy to make in natural situations, and that could be a reason why life may be very common in the universe. Now, will it be little green people in spaceships? Probably not, but could it be bacteria on other planets? Absolutely, and even that would be a very astounding discovery. So proteins are made of amino acids. There's 20 kinds that living things use. We put amino acids together, and these are called peptides. So we have a polypeptide. Dipeptide would be two together, tripeptide, tetrapeptide, but basically poly means many peptides. And these letters actually represent, it's a code for different kinds of amino acids. Now, every time you link two amino acids together, and you would do that by popping off a hydrogen and popping off the oxygen, and then the hydrogen, which should be here, you connect them together through dehydration synthesis, you're making a peptide bond. Peptide bonds are the covalent bonds between protein subunits, which are the amino acids. Like I said, there's 20 amino acids. These are the 20. They all have different groups on them. So this is valine has two hydrogen and carbon groups coming off of it. This is a non-polar amino acid, at least the subunit. This part is still polar, but this part is non-polar, this unit coming off. Also non-polar. Also fairly non-polar, although there is a nitrogen here and a hydrogen. That's tryptophan. That's supposed to be the one that makes people sleepy at Thanksgiving from eating the turkey. Glycine we talked about, just a hydrogen. Tyrosine has an OH, a polar part of that molecule. Asparagine has a nitrogen and an oxygen, making that polar. Sulfur hydrogen, not really very polar. Glutamic acid, very polar. Aspartic acid, very polar because of the oxygens. With the nitrogens, these are polar. And then we have the non-polar ones, phenylalanine, very non-polar. So someone who has a condition called phenylketonuria ends up not being able to break this down in a proper way, and that's actually a fairly severe genetic condition where someone has to limit the kinds of food they can eat. You'll see a lot of times warnings that certain foods contain phenylalanine. And then these letters at the bottom, that's the code. So valine, alanine, phenylalanine is an F. These are the codes, so when we look at a chain like this, we could actually tell which amino acid is which in the code of the protein. So the structure of proteins is fairly complicated, but that's how proteins work. So the basic sequence of amino acids, and we'll work on this a little bit in class. The basic sequence of amino acids, that's the primary structure. That's the chain of amino acids. Then, because of those repeating units and the shape of these units, they assume certain shapes. They may assume an alpha helix, so kind of a nice curvy, curly, staircasey kind of a design, or a pleated sheet where it has much more jagged ridges, but it does almost have a staircase pattern also. Then you'll have certain amino acids, because they either have nonpolar or polar portions on them, they'll be getting attracted to each other. So they'll start to fold and coil in on each other in different ways. So this alpha helix now starts to fold around different portions of the molecule, and those could be like phenylalanine with that big carbon and hydrogen group coming off, or valine, or one of these nonpolar ones. The nonpolar subunits will find each other and want to stick with each other, whereas the polar ones would either want to stick with water, or they might want to stick to each other as well, depending on the environment where this protein is. So that's the tertiary structure. So primary is first, secondary is second, tertiary is third. This is where we form kind of a tangle in the protein, but it's supposed to form this tangle. That's how the protein actually works. And then quaternary, that's the fourth. That's where more than one protein chain comes together and makes a much more complex protein. So one of these proteins, very complex, is hemoglobin. Hemoglobin is made of 4 different proteins all twisted together with each other in order to carry oxygen throughout your body. Function of proteins, they form many structural parts of cells, muscles, excuse me, and the tissues that connect organs together. Enzymes are also incredibly important proteins. They make certain chemical reactions, such as breakdown of sugar, possible within the human body. Sugar would generally burn at a very high temperature, too high for living things to be able to handle. However, the enzymes that are inside the human body enable humans or other living things to break down glucose at much lower temperatures than would normally be required. And that allows us to get energy in a safe way without burning our bodies. That's thanks to these enzymes. So we find proteins in meat, fish, legumes, nuts, milk, eggs, grains, and soy products. Most things have proteins in them. Meats, things that come from animals, tend to have more protein. Fish, legumes, those are beans, nuts. But the things that come from animals, eggs are primarily protein with a little bit of cholesterol and some other nutrients. Milk has a lot of protein. Meat, fish, lots and lots of proteins. Certain grains have some proteins. Wheat has some protein in it. Soybean things have a lot of proteins in them. We as humans cannot make 6 of the amino acids. We call those essential amino acids because our diet needs to produce them. And we get some of these essential amino acids from things like beans and rice and nuts and fish. We get proteins that our bodies cannot produce, and we need to get them from our food. Now, the Burette solution is the way that we can tell if a solution contains protein. It will change blue or purple if the protein is there. All right, moving on to carbohydrates. Carbohydrates are made of simple sugars called monosaccharides. We can see here we have carbon, 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 carbon. One more carbon, that's C6. H, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. H12. O, 1, 2, 3, 4, 5, 6. I think I counted them right. C6, H12, O6. That's glucose. Most of these sugars end in an OSE, so we have glucose, maltose, galactose. All of these are simple sugars. They're monomer. They're called monosaccharides. You put 2 sugars together, you get a disaccharide, and then we get polysaccharides, which are many of these glucose, or another sugar strung together. So those would be starch. Starch is something our bodies can digest, made of glucose, which our body runs on as an energy source. Cellulose is also made of glucose, but as we saw in class, the linkages between cellulose molecules means that our body can't digest it. Very, very few things can digest cellulose. Some bacteria can, a couple of other microorganisms can, but very few are able to. Chitin is very similar also, a structural polysaccharide that's used in insects and other animals with an exoskeleton. It's very hard to digest also. Very few creatures can digest this. Glycogen is an energy storage molecule in humans. It's another example of glucose molecules stuck together that our bodies can digest. Now, starches we use for energy storage. Animals can digest starches. Lots of plants also use starches. When we eat those starches, we're able to digest them. So what happens if we started to digest them? We would get a complex carbohydrate like a starch, and then we would break it into smaller pieces. These are disaccharides because there are two sugars stuck together. And then we would break those disaccharides into monosaccharides like glucose. Now, certain carbohydrates are difficult to digest, cellulose being one, chitin being another. Those are ones that are used for structure, very hard to digest. Therefore, animals like cows and giraffes and camels and goats need a special structure in their stomach to hold bacteria that will digest the cellulose for them. We have some animals like horses and rhinos that make a big chamber for bacteria in their large intestine where they can break down the cellulose. And then rabbits do the other thing. They eat it entirely. The very end of their intestine is where they have bacteria to break it down. They poop it out. They re-eat it. That's how they get the nutrients that the bacteria have broken down for them. But horses don't need to do that, and cows don't need to do that. What cows will do, though, they will get that bacteria into that. They'll get that food, that grass into the first chamber, and then they will pull it back up into their mouth and chew it a second time so that it goes all the way through. Horses don't need to chew their food a second time, but they chew it very extensively the first time. They have very, very good teeth for chewing. Then it goes back to the rear part of their gut where they have bacteria that will break down the cellulose. And then the rabbits, they chew it fairly well, but not enough for it to be working one time through. Rabbits need to eat their poop the second time through in order to make sure that they get the nutrients and the glucose out of that cellulose. We find carbohydrates in a lot of different substances. Simple carbs, the simple sugars, the simple monomers, well, obviously in candy, those are disaccharides. Sucrose is one glucose and another sugar called fructose stuck together. If you ever see high fructose corn syrup, same thing, it's sucrose. We find high fructose corn syrup in lots of sodas. We find other simple sugars in fruit, vegetables. We find them in milk. We find lactose in milk, which is that's glucose and galactose stuck together. It's another disaccharide. These are quickly digested. They give you a lot of energy. Complex carbohydrates, starches and such are found in pasta, bread, potatoes, beans, corn. They take a longer time to digest, and they will provide longer energy because your body has to break down these carbohydrate molecules, these long polysaccharides. Your body takes time to break off each of those polysaccharides and break it down into as an energy source. We'll talk about how it breaks down as an energy source later on. Indicator test for carbohydrates. We have something called Benedict's solution, which when you put a blue solution of Benedict's with carbohydrates, it'll turn orange, green, or brown. We also have Lugol's solution or iodine. That's the stuff, maybe if you have a cut, you end up putting iodine on it. It's reddish brown liquid. You put that into a starch or a carbohydrate solution, and it will turn blackish purple. And there's an example of the iodine turning black or purple in the presence of a carbohydrate. Now, the last polymer are the nucleic acids. The structure of the nucleic acids, they're made of nucleotides. Now, this is the most complex of the monomers because, look, we actually have this nitrogen-containing base. So there might be a ring or two here with nitrogens and carbons and hydrogens stuck together. Then you have a sugar. This is called a ribose or a deoxyribose sugar, which has 1, 2, 3, 4, 5 carbons in it, not 6 like glucose. And then the phosphate group. So right now, the thing that'll tip you off to something being a nucleotide is if it has a phosphorus. If it has a phosphorus, chances are it is this, especially if it has this structure. If you happen to see a phosphorus with a long chain of carbons and hydrogens, well, then you have a phospholipid. But in this case, we have a nucleotide. The polymers, the most common ones are DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. DNA is a double chain, where they each go along like a staircase around each other. And RNA is a one-strand molecule. So these have very different functions within an organism. But just to know, DNA almost always is double-stranded. RNA is almost always single-stranded. Now, the function of nucleic acids, they carry genetic information. The code to make any living thing is in DNA if it's anything above a bacterium. Now, some viruses, even though we can debate whether they're alive or not, some of them have RNA. That's another reason why we don't exactly agree that viruses are alive, because a characteristic of life is that it has DNA. And if it has DNA, it's alive. Although, again, it gets tricky because some viruses have DNA. This is why viruses are a very unusual area talking about living things. Also, nucleic acids help to build proteins, but the proteins are all coded by the DNA. We'll talk more about the code of DNA with one more video about macromolecules. Some people, many scientists, think that actually RNA was the first molecule that could make a copy of itself, which is one of the basic characteristics of life. So we may have had kind of a soup of where these little nucleotides were in the oceans or near a deep sea volcano or in somewhere else on the earth. And then they started sticking together because of an energy, maybe lightning, maybe heat from a volcano, caused them to start sticking together. From there, they made longer chains. And then some of these chains may have been able to actually make an extra copy of themselves. RNA can actually make a copy of itself. And then over time, these RNA self-replicating molecules were able to find some sort of a lipid containing bubble. They were able to get inside, and they were able to now make copies of themselves and then get so many copies that this lipid would actually split and we'd have two cells. That would be the first evidence of replicating life. And then over time, DNA, which is a little bit better at coding than RNA, would have formed proteins, which are better at building parts of the cell, would actually take over. But RNA could have been the first original source of life. We call this the RNA world hypothesis. It's a possible way that life emerged on Earth or somewhere else in the universe and was transported to Earth. So the thought is that RNA may have been this first self-replicating molecule, the first molecule to make a copy of itself, which meant that it was meeting the first characteristic of life. We don't really have a great explanation for how life formed quite yet, although this appears to be a possibility. Now, did this happen on Earth? We don't actually know, because the conditions for life on Earth, as soon as life could exist on Earth, we see life on Earth. So either life arose very quickly, or life maybe arose somewhere else, possibly Mars, and then was transported to the Earth. That's another possibility. Or somewhere else in the universe. There could be self-replicating molecules anywhere in the Earth. It just so happened that Earth was a favorable place for them to end up. So we find nucleic acids in any other living things, from bacteria up through from bacteria up through humans and trees and mushrooms, because that's our code. That's another way we know all life is related. We all use the same code. A bacteria uses, in the hot springs of Yellowstone, uses the same code as a human does, or a maple tree does, or a yeast cell, or a fungus, other fungus of some kind. They all use the same code. So really, any living thing we can find the building blocks for DNA. Now there's one. It's kind of not a thing that we'll do, but this diphenylamine test will turn light blue to dark blue if nucleic acids are present. There's another way of finding nucleic acids, isolating DNA, which we'll actually do an experiment where we isolate DNA. Another way where you can actually truly see it. Now, in all of these macromolecules, we eat them. We break them back to their monomers. So that's like taking apart a brick building. And then what do? We build them into what we need. So the subunits are the same. The amino acids that come from a plant are the same amino acids that a human uses to build proteins. So we break down those amino acids and then we build them to what we need. Everything in our body has been taken from something else, a plant or another animal, digested, broken into those monomers, and then built up into the monomers that we need. So the carbohydrates in us, they came from the food we ate. We built them into what we need. The proteins in us, we took them from other living things. We broke them down into the subunits and built them up into what we need. The DNA from, let's say, a mushroom that you ate, we break apart those nucleotides and then we build them into human DNA to code for a human rather than a fungus. And just to remember, when we digest carbohydrates, we break them down to the simple sugars like glucose. Lipids, we break them down to the fatty acid and glycerol. Remember, we break them down by hydrolysis. We build them up by dehydration synthesis. Dehydration synthesis, you will see a water molecule come out. Hydrolysis, you'll see a water molecule go in and become part of the monomers. Proteins are made from amino acids, and then nucleotides make up the nucleic acids. And that concludes this presentation. Thank you.